Good morning, everyone. Let's get started. There are a couple questions that I missed last class for the lecture number 6. We will do those first. Okay, so if you can take out your study guide from lecture 6, I missed question 13, and then I said we would do the application question today for that particular lesson. So lesson 6, lecture 6, number 13, lists 3 main functions of the cerebellum. So just to remind you, the cerebellum is at the back of your brain. We will review the structure of the brain in a second, but let's just fill this part in first. Okay, so the 3 functions of cerebellum. The first function of the cerebellum is that it is the location for stores skilled memory. Okay, so things like riding a bike, playing the piano, playing any instruments, typing, all those skill memories are going to be stored in the cerebellum, and those are very, very persistent. They do not go away. If you don't ride a bike for 10 years and I give you a bike, you can still ride it. Okay, it might take some time to get to be good at it again, perhaps, but then you will not forget how to do it. And so all those skill memories are stored in the cerebellum. Another function of the cerebellum is that it maintains balance and equilibrium. Okay, so the cerebellum actually receives multiple inputs from various sensory structures that would give it an idea of the relative position of your body parts, and then it would compare it to a reference of what those body parts are supposed to look like or at what position when you're doing certain activities. So for example, if you are sitting, I assume most of you are sitting right now, then there is going to be like a reference picture in your cerebellum of what sitting should look like. Okay, so sitting would be something like this. Okay, so your center of gravity is low, and then your legs are this way, and then that's the reference of what sitting should look like. And then you're going to be receiving sensory information from the eyes, from the ears, and from the proprioceptors in your muscles, and then the cerebellum will look at your actual position and compare it to the reference. So the actual position is going to be the same. So what happens is, if all of a sudden the leg of a chair breaks and then you fall like this, okay, falling like this, then all of a sudden the cerebellum will know that your actual position is now not the same as the reference. Okay, sitting is supposed to look like this, but all the sensory signal coming in from various organs tells you that you are not looking like that, you're falling. Then the cerebellum will try to readjust your body to go back to that position. I mean, in the case of the falling chair, there's nothing you can do. You're going to hit the floor, okay? This would be more of a situation when you're walking, okay? So let's say walking, then cerebellum knows your walking is supposed to look like this, swinging your arms, moving your legs. And then if you trip on a rock, okay? If you stumble upon a rock and you almost fall, and then you regain your balance, right? That is the function of the cerebellum. Okay, so cerebellum would try to maintain balance, maintain equilibrium. If you lose that balance, it's going to try to regain that balance. Sometimes, most of the time it works, sometimes it fails. Next one, the last function of the cerebellum is that it will ensures smooth execution of movements. Okay, So think about someone having a basketball, and then the basketball net is here. If this person is going to dribble the ball and then do a layup, right? It's like, trying to shoot the ball in by jumping and putting it in there. So the action of walking all the way there while dribbling the ball up and down, you are able to execute that in a seamless transition because of the cerebellum. Okay. Same thing. If you imagine you are walking up the stairs, okay, you're able to walk up the stairs virtually without thinking about it, right? Shifting your balance as you go up the stairs, moving your legs, swaying your arms, all those things, being able to do it in a smooth, seamless fashion. That is the job of the cerebellum. Okay, if you damage the cerebellum, then you're gonna have like jerky movements. It would be stumbling around as if the person is drunk. So that's basically the cerebellum. Okay. Now I am going to show you some of these models to just refresh your memory, because I know last week you guys had a lot of tests and whatnot, so you might not remember a lot of the stuff that we went through. So I'm just gonna take maybe about 10-20 minutes to go through some of the key features of the spinal cord and the brain. Hopefully this will help you refresh your memory a little bit. Now, these models are all available on eCentennial. Okay, so if you just go to eCentennial, let me quickly show you where it is, because I don't think a lot of you have had a chance to go through them. So in your core shell, contents, we are doing unit 2 and nervous system, part 2. If you go to the studying resources, you have your study guide and stuff like that, video resource, coloring sheets, and then there's a 3D model. So if you click on one of these things, it will take you to the 3D models that I'm going to be showing you right now. So here is, I know it says spinal cord here, but can somebody tell me what am I looking at? What is this thing? What is this thing that
vertebra?
That is the vertebra. That's right. So vertebra is your backbone, right? Basically, and they stack one on top of each other. We'll learn more about the features of the vertebra later on. But this one would be your lumbar. Remember we talked about this, right? The cervical, the thoracic, and then the lumbar, right? So this one would be one of the lumbar vertebra, okay? And this is the back. And this is the front. I mean, if you just, I can't really show you on the camera, but like maybe you can see, if you run your hand like along the middle of your back, right, you feel these little ridges, right, little knobs, right? What you're feeling is essentially this part of your backbone, okay, the spinous process we call it, okay. But that's not what we're focusing on right now. Okay, if we look at the middle of the backbone, okay, if you look at the middle of the backbone, you will see that there is your spinal cord. There's your spinal cord. Let me see if I can turn off the label. There's your spinal cord. Okay, and if I look at the side, you see there is the spinal nerve coming out. Okay. In a little opening that is formed between two of these backbones. Okay, so you cannot really see another one, but there's going to be another one on top and another one below. And on the side where the two kind of connects together, there's a little opening on the side for the spinal nerve to come out. Okay, and between the backbone, there is usually a disc of cartilage that separates them. That discs can experience wear and tear as we age, and then the space between the two vertebrae is going to get thinner and thinner, and then it can come down and kind of pinches on the nerve, right? So that's called pinched nerve, and people who have that would experience some kind of tingly numbness sensation in part of the bodies, depending on which nerve is being pinched. Okay, so I'm going to delete the vertebra, and you can see here is our lovely spinal cord, and I'm going to rotate it so you can see it. Labels are kind of in the way all the time. Okay, so here we go. Again, you can see that this looks like a butterfly, right? Okay, this looks like a butterfly. So the bottom of the butterfly is actually going to be at the back of the body, and this is going to be at the front of the body. Okay, so let me just reset it for you and take out the bone again right here. Okay, so you can see the dorsal side. Dorsal means the back, ventral means the front. Okay, so in this spinal nerve, last time we talked about the fact that there is going to be a sensory neuron coming in. Okay, and sensory information is going to be entering at the back, right? And it's going to be making a synaptic connection with an interneuron in the gray matter, right? Okay, so from there, it's going to connect with a motor neuron, and the motor neuron is going to exit at the front and then join with the sensory neuron. So the motor neuron and the sensory neuron here is bundled together to form your spinal nerve. Okay, and then it just splits, right? For the sensory information to go in at the back and the motor information to go out at the front. Okay, so you can see in the spinal cord, the gray matter is in the center. That's where your interneurons are going to be. That's where you're going to be doing the processing power, right? For a lot of the reflexes. You're going to have the white matter at the back. So can somebody tell me at the back, what kind of tracks are we going to have? What kind of informations are carried by the tracks that are on the posterior side, on the dorsal side? Anybody?
Would it be the ascending tracks?
That's correct. That is the ascending tracks going up to the brain, right? And what kind of information, Colleen, does the ascending track convey? Remember, or someone else? Yeah, go ahead.
It's telling the brain basically to make the decision.
Yeah, and the kind of information that is transmitted in the ascending track, as Stephanie mentioned as well in the chat room, is the sensory information. Okay, sensory information. So sensory information, let's say from the hand, if you touch something hot, that's gonna go to your spinal cord through the spinal nerve, enters at the back. Part of that, it's gonna continue the reflex circuit out at the front, but part of it will now be passed on to the ascending tract, which will then go up to your brain. Okay, and then once your brain has made a decision of what to do, those motor information will now be coming down at the front of the spinal cord through the descending tracts. Okay, and then they will exit at the appropriate section. Okay, so there is the spinal cord. Now, of course, there is that little hole in the middle that we keep on talking about last class. Again, anybody remember what that hole is? What is it called? What is it for? Why is it there? Anybody? That is the central canal. Okay, central canal is where the CSF is going to be traveling down, right, from the fourth ventricle at the top. It's going to go down in the middle of the spinal cord within this central canal until it gets all the way to the bottom. Okay, a couple more questions just to see, where you guys are at. What is this little gray halo film that that surrounds the spinal cord here? What is that?
Meninges?
Yes, that is the meninges, or meninx, if you look at the individuals layer. Okay, so you have the dura mater, you have the arachnoid mater, and you have the durovenous, the pia mater. Okay, so that's it. That's it for the spinal cord. Any question? This is kind of like a review, right, of what we learned last class. There's another model here that I want to show you. We talked about this last class, but let's just go through it one more time. Because why not? Okay, so here we have the skull, right? Okay, maybe I'll just leave the label. Here is the skull, right? That's like your cranium, right? Okay, and outside of that, that's like the skin of your scalp, right? There's something called periosteum, which is just like a connective tissue that covers all bones. Don't worry about that. We'll learn that in the bone unit. Okay, but underneath the skull, right, or if we're looking at the spinal cord underneath the vertebrae, you would have the dura mater. So here, this is the dura mater. I should remind you that the dura mater is actually two separate layers, okay, that are usually fused together, but sometimes they are going to open up at certain locations. So here is like, two pieces of dura mater, they are usually stuck together like that, but at certain location, at certain places, they're gonna open up. I'm gonna try to open this. They're gonna open up like this, right? And have a little opening in the middle, a little hole, okay? That space in the middle, right? That is the dural venous sinus. That is where you're going to be draining your excess CSF. And one of the locations of the dural venous sinus is like right here, right? In the middle, okay? So everywhere else, they are going to be fused together, the dura mater, but up here, it's going to open up like I showed you. And that space in the middle, that's where you will drain the excess CSF. Underneath the dura mater is going to be your arachnoid mater, okay? And the arachnoid mater actually has a subarachnoid space, right? You can see all these fibers. They kind of resemble spider webs. That's why it's called the arachnoid mater. Arachnoid means spider, okay? So then this subarachnoid space has a lot of empty spaces for the CSF to fill up, and the CSF, of course, create a little bit of a buoyancy as well as cushioning capability, right, to protect the brain, right? So that if the brain moves, it will not crush into the skull because of this buoyancy, right? This cushioning capability. And then finally, there is the pia mater. It will follow the grooves and the bumps of the brain, and it, is pretty much completely stuck to the surface of the brain as well as the spinal cord. Okay, and here is the brain. You have the gray matter on the outside, and you have the white matter on the inside. The gray matter on the outside is called the cerebral cortex. Okay, that's where your interneurons are going to be found. If you remember earlier, I showed you the spinal cord. The gray matter is in the middle for the spinal cord, but the white matter is on the periphery. All right, it's kind of opposite. Now, there are exceptions. Even though most of the white matter is inside the brain, there are centers of gray matter that are found on the inside as well. And one exception would be the reticular formation, right, or the thalamus, which we'll talk more about in a bit. Any question? Just want to see if anybody needs clarification on anything. Okay, so that is done. Let me close that, close that. Here is the brain. And let's just go through some of the brainy stuff here. Okay, which half of the brain is this? Is this the right half or the left half? Anybody?
Is that the right?
Right.
That is the right. We're looking at the right half of the brain, the right hemisphere, right? The right hemisphere, and we're looking at it from the left. Okay, so this is the back of the brain. You can always tell because there is this cerebellum here, okay? And then this is going to be the front, okay? If you see this dangling thing here at the front, right? That's a pituitary gland. When you see that, that's pointing at the front, and then the cerebellum shows you at the back. So let's go through some of the structures here. Now, I am going to just point to stuff, and I'm going to ask you what it is. Okay, if you know it, you can type it in the chat. and then, participate a little bit, okay? Or you can say it if you want, and then I'm going to describe what it is. Okay, so let's start with something easy. I talked about this like at least 5 times last week. What is this band here? This band. Anybody?
Corpus callosum.
Corpus callosum. That is correct. Okay, so maybe someone can type that in the chat if you want, just so other people can see it. I cannot write on this app right here, so as much as I want to label it, I won't be able to do that. Okay, so that is the corpus. If I click on it, it's right here. Okay. Corpus callosum. Okay, the corpus callosum, it's a bundle of white matter. Remember, white matter is basically axons that are myelinated. Okay, so they are primarily for communication. So the corpus callosum is responsible for communication between the right hemisphere and the left hemisphere. The two halves of the brain needs to talk with each other constantly. Okay, so information flows from one side to the other, and they do so most of the time through the corpus callosum. Okay, so sometimes people have to undergo surgery to sever part of the corpus callosum. It's one of the extreme treatments for people who suffer from a lot of seizures, okay, epilepsy. Okay, so one form of epilepsy is that, first of all, what is epilepsy? What is seizure? Basically, it's uncontrolled firing of neuron. Okay, so that those action potentials go all over the place, and it causes the person to seize. And so if the seizure is spread from one side of the brain to the other, right? One of the ways to kind of prevent that from happening, scientists have discovered, is to cut part of the corpus callosum. Surprisingly, after cutting the corpus callosum, those people are virtually okay. They don't lose any significant function. Okay, so that is quite interesting. Underneath the corpus callosum, okay, this thing right here. Anybody know what that is? This structure. It's actually a hole. Okay, I can stick my hand into it, and then I can feel a really big space. Rochelle said it is the lateral ventricle. That is excellent. Okay, now I'm going to delete a bunch of stuff to show you what the lateral ventricle looks like. Okay, wish there's a quicker way to do this, but here it is, and that's why this is a good program to it's free, right? And it's already on eCentennial. You can play around with this, and you can learn a lot from it, actually. Okay, and you can see there's a lot of stuff that we didn't talk about. Okay, which is fine. This is the introductory course, right? So you can see the lateral ventricle is actually a C shape, right? Remember, if we look at it from the front, it kind of looks like a horn, right? That we have, and that is the lateral ventricle. Within the lateral ventricle, we talk about, and actually all the other ventricles, there are these spongy structures, right? They are called the I'll type it on the in the chat. It's called the choroid plexus, and the choroid plexus is where you will produce the CSF, right? The cerebral spinal fluid. Okay, so there is the ventricle, and from the ventricles, it's going to drain into the third ventricle. Okay, so you can see here's the third ventricle. Okay, from the third ventricle, it will go down the cerebral aqueduct into the fourth ventricle. And then you can see here, you can see from the fourth, it will continue to go down. Right? This is your medulla oblongata now. There is a hole at the bottom of your skull where the medulla oblongata will come out, and that's going to become the spinal cord. Okay, can you visualize this? I mean, I don't have the spinal cord on this diagram in this model, but the spinal cord would be like right here. Okay, coming out at the base of your brain. Okay, so you can see that this fourth ventricle is going to continue to go down, and it's going to connect with the central canal that we were talking about earlier, right? That little hole in the middle of the butterfly of the spinal cord. Okay, so where would you say the fourth ventricle is? The fourth ventricle is between the cerebellum. Remember cerebellum, the first thing we talked about today, right? Maintain balance and equilibrium, store skill memory, as well as ensure you have smooth execution of your movements. Okay, in front of the fourth ventricle is going to be this structure. Anybody can tell me what this structure is? There are two things here, actually. There's a little bulge here. That's called one thing. And then on top of it, there's another thing. Anybody? Now you have to, if you are unfamiliar with this, you have to study a little bit for the test, okay? Because on the test, I will show you a brain, something like this, and then I will be pointing to it. Okay, I'll be like, what is that? And then you have to know. You have to be able to choose the correct answer. Okay. Thank you, Joan. Joan said the medulla oblongata. That is a good try. Okay, the medulla oblongata is actually right here, and that's one of the two things. And then the other one is pons. That's right. Okay, so pons here and medulla oblongata, both a good try. I shouldn't say good try because it's actually correct, right? Both are good answers. So that's where the fourth ventricle is. Okay, up here, that's the midbrain. So midbrain, pons, medulla oblongata, they make up your brainstem. Okay, they make up your brainstem. So in the medulla oblongata, while we're talking about it, there are several reflex centers, okay, involving cranial nerves. So your coughing reflex, like if you have something stuck in your throat and then you want to cough, right? That's located in the medulla oblongata. There is the respiratory center, right? Vomiting reflex, swallowing reflex, all those things are found in the medulla oblongata. So can somebody, anybody know what do we take when we have a cough? Well, cough syrup, right? You might think, but what are some of the active ingredient in cough syrups? Anybody remember or know about it? Start with a C, the word I'm looking for. No? Codeine, anybody heard of codeine? No, maybe? Okay, codeine is a very mild opioids, okay? And it works by suppressing the medulla oblongata, okay? Because that's where the coughing reflex is, right? Okay, so you probably have heard of the fentanyl crisis. We talked about this earlier in lecture 5. That is also an opioid, right? But much more potent. And it also acts on the medulla oblongata, much like codeine does. But when you OD on it, it suppresses the medulla oblongata so much that it causes your respiratory center to not work. And that's how people OD on fentanyl dies. They stop breathing, essentially. Fortunately, there is a way to fix that if discovered early. There's an antidote, right, called naloxone that can be administered, and it will reverse the effect of fentanyl if given quick enough. Okay, let's reset the brain and bring everything back. This is the third ventricle. If I delete the third ventricle, you will see that this is the right thalamus. Okay, so thalamus is the gray matter, and it is like a sorting and filtering center of the brain. So all the sensory information is going to be going through the thalamus before it gets projected to different parts of the brain, except for smell. Okay, so your nose is right here, and the temporal lobe is the lobe that is responsible for processing smell. So it would just go directly to the temporal lobe without passing through the thalamus. Okay, everything else is going to pass through thalamus. So if you have remember we said touching a hot surface, and then that information is going to go up the spinal cord at the ascending tracts at the back. So all that information will eventually go up the brainstem, and then it will go to the thalamus, and the thalamus will project it onto your primary somatosensory cortex, okay, which is, if you look at the lateral side of the brain, which is this piece right here, okay. Postcentral gyrus is talking about this bump located on the postcentral gyrus would be the primary somatosensory cortex. Okay.
Question.
Yes, please.
Just above the the pituitary gland, is that the hypothalamus?
That is correct. Okay, so let's get to that right here. This again is the thalamus, okay, for filtering sensory information, as I just mentioned. And also it makes up the walls, the sides of your third ventricle, right? Okay. If you go down the thalamus, you have the hypothalamus. Hypo means below, right? Hypothalamus is the master gland of the body. It takes care of a lot of the autonomic functions, right? We talk about sleep-wake cycle, your circadian rhythm, right? You might have heard about it, right? Like when do you go to sleep, when you wake up, right? That rhythm, right? That's controlled partly by the hypothalamus, your water balance, your body temperature, right? And a bunch of other stuff that you will learn in the last lecture of the course. The hypothalamus works closely with this thing here. That is the pituitary gland. It's separated into the anterior pituitary gland and the posterior pituitary gland. Each of them would have a bunch of hormones coming out from it. Okay. So that is pretty much that. I mean, there are some other structures that are inside that I cannot really show you without. I mean, it wouldn't really help because they all look the same here. It's much better to just look at the PowerPoint for that one. So for example, if I cut out a bunch of other stuff here, you will see that is where your amygdala is, okay? Anybody know what the amygdala does? We talk about that in class. Anybody remember amygdala, right? And then there is your hippocampus, right? It's connected to the amygdala. What does the amygdala do? Anyone? That's right. Amygdala makes you have emotions. Thank you, Joan. So if you feel happy, sad, excited, embarrassed, frustrated, right? All these emotions come from the amygdala. It is all part of your limbic system, right? The limbic system is the reward system. So if you do something that makes you feel good, right? You will remember that feeling and you will want to do it again in the future. If you do something and it hurts you, it makes you feel scared, right? You will try to avoid that in the future. Okay, that's part of your limbic system. And part of the limbic system is this structure right here, right? Let me delete some more. You'll be able to see it right here. That is the hippocampus, okay, which actually means seahorse. I don't care what they say. This looks nothing like a seahorse, but the person who discovered it's like, oh, seahorse, let's call it hippocampus. So hippocampus is for long-term memory, right? Things that you're going to remember pretty much for the rest of your life. And it's also part of your limbic system, okay? So once again, the kind of like the factual stuff, right? Those kind of memories are stored in the hippocampus, but skilled memories are stored in the cerebellum. Any question before we move on? It's a lot of stuff, okay? This is probably one of the hardest thing of this unit, okay? And since your test is in 2 weeks, it's very important that you start studying, preparing as soon as you can. Okay, what else do we have? See, there's an other brain here. I'm just gonna quickly go through a few simple things and then we're gonna move on. Okay, this is coronal cut, right? Frontal cut. And you can see this is the right side, this is the left side. We're looking at it from the front. What are these two big ovals here? Anybody? Two big ovals here. What are they? Nope. Okay, that's the thalamus. Okay, left thalamus, right thalamus, and between the thalami, what is this here? If you have been paying attention, you should know what that is. Two thalami in between them. What is that? That is the third ventricle. Okay, third ventricle. So you can see you have your lateral ventricle here, and then it's going to drain into the third ventricle. Okay, so other stuff that we see here, that's the corpus callosum. okay, and it actually spreads throughout the within the hemisphere here. Okay, you can see that's the pons, and then that's the medulla oblongata. Okay, hippocampus, right? Okay. You cannot see the amygdala. If this is the hippocampus, the amygdala would be like somewhere out here at the front. Okay, and then there are a bunch of other stuff that we did not talk about, which is fine. Okay, we cannot possibly go through everything. Now, I do want to show you the insula here. Okay, the insula is like the fifth lobe. Okay, that's where you will process taste, gustatory information. Okay, so that is the parietal lobe here, and that is the temporal lobe. And then you can see that part of the insula is right here. Okay. So play around with those things when you have time. Make time for it, and then it will help you, remember these things. Also, a good idea is just to go on Google image search up like brain diagrams, or search up like real brains, and then try to label as many parts as you can, right? So again, corpus callosum here. You can see the lateral ventricles here. That is your thalamus, right? So label as many things as you can. The more you do this, the better you will be. Okay. We still have 2 application questions, but let's start lecture 7 now. Okay. If we have time, we'll come back to this. If not, we will do it, during the review next class. Okay. Actually, you know what? Now that most of you are here, let's just talk about what is going on for the upcoming weeks. We are actually in the 7th week. No, we are in the 8th week now, right? Okay. So this week we will be doing the sensory structure. We're doing it right now. And then next week we will do the digestive system. Okay, so digestive system should not take too long. We should be able to finish maybe hour or hour and a half, and then we will also take up the lab and then do the test review as well. Okay, so all of you know where to find the labs now, right? There's a link on eCentennial that you can get it. So start working on that, perhaps this week, and then try to finish it before you come, so we can take it up. And then you will have the lab quiz. The lab quiz will be available for 2 days, just like last time. It would be 2 attempts, and the best score is going to count. Now, you might or might not know this. November 12th is the last day to withdraw without academic penalty. So far we have completed. By the time you finish lab 2, we would have completed 45% of the course. Okay, so you have your quiz 1, quiz 2. That's 20%. Test 1. That's another 20%, so 40, and then 2 lab quiz, right? So that's 45%. Okay. On eCentennial, I should probably tell you this. If you go to the course homepage again, there is if you go to content, and if you go to useful resources, there is a grade calculator. Okay, so if you click on that, you can download. You have to download it for it to work. It's an Excel file that I created. Okay, so you download it, and then this tells you how to use it. Basically, you can key in your grades, right, for quiz 1, quiz 2, lab 1, lab 2, and test 1, right? And then it will tell you what your current grade is. Okay, and then you can key in what final grade you want. Okay, let's say you want a 70 because that's the minimum requirement for the nursing program, for example, right? Then the spreadsheet will tell you what you need to get for the remainder of the course in order to get the 70. Okay, so you might want to play around with that so you have an idea of where you stand. Okay, so the last day to drop without academic penalty is November 12th. It means if you are failing the course right now and then you're not really sure if you can pass, obviously talk to me. Okay, you can talk to our advisor, Martin, and then sometimes the best option is to drop the course now so it doesn't show up on the transcript and then retake it later. Okay, but there are repercussion if you do drop a course. So before you make any decision, it's best to talk with your teachers or again, our advisor, Martin. Some of you know Martin Rosowski. He would be able to give you some guidance as well. But contact me if you're concerned. And then you will have your tests, okay? The tests unfortunately happens after the drop day. So you wouldn't really know how well you do, but it's the same thing, right? Test 2 is similar to test 1. Okay, questions? No questions. All right, so let's do this. Sensory special senses. Okay, so special senses talks about the various type of receptors we have throughout the body, which will help us detect different triggers, different changes in the environment, external environment as well as internal environment. So starting with the first one here, mechanoreceptor. Actually, you know what? Let's read the definition and look for the answer. Is much easier that way. This receptor is stimulated by damage or oxygen deprivation to tissue. Okay, that would be the nociceptors. Okay, nociceptors is a special word for pain receptors. So this is basically pain receptor. There are two types of pain receptors, right? There is the visceral, right? Visceral. I should write down visceral nociceptor. As opposed to somatic nociceptors. Okay, anybody want to tell me what the difference is between visceral versus somatic? What kind of
one outside, like skin and maybe skeletal muscles, and the viscera, like viscera's internal organs.
Exactly. Thank you, Mohammed. So visceral is internal organ, right? Like your kidney, if you have kidney stone, it hurts. That's going to be related to the visceral. Okay, surface damage is from somatic. Okay, so things that you have voluntary control mostly. Okay, skin, muscle. Next one, stimulated by changes in external or internal temperature. That is the thermal receptor. Okay, stimulated by changes in pressure or body movement. That would be mechanoreceptor. So change in pressure would be like, poking your skin with the blunt end of the pen as opposed to the sharp end. It feels different, right? So different mechanoreceptors will pick that up. Body movements, okay? If you move your head like this, okay, you can feel it, feel a little bit dizzy maybe if you move it a couple more times. There are mechanoreceptors in your ears that detects that, okay? So some mechanoreceptors are triggered by changes in pressure. Some mechanoreceptors are triggered by body movements. Stimulated by light energy, that would be photoreceptors. Okay? You have photoreceptors in your eyeballs. There are three types of photoreceptors. Well, okay, there are two types of photoreceptors. The cone cells for detecting color and the rod cells for detecting in low light environment. And then there are three types of cones. One for red, one for blue, and one for green. Stimulated by changes in chemical concentrations of a substance, that would be chemoreceptors. Okay, so you have chemoreceptors on your tongue, for example. Okay, identify the parts of the eye in each of the statements. All right, so I will quickly go through the eyeball with you, and we'll fill that in. So here is the eyeball, and it's cut open, so you can see the inside. The eyeball has three layers. Okay, the superficial layer is the sclera. Okay? That's the white part of the eye right here. Okay, sclera. That provides attachment sites for muscle. Right, you have a lot of muscle connected to the eyeball, so your eyeball can move left, right, up, down, and even rotate as well. You roll your eyes at someone. Right, there is a muscle for that. So that is the sclera. Now the sclera is going to become the cornea at the front. Okay, I'm not sure why I cannot. Oh, there it is. You cannot really see it being highlighted. Okay, but the cornea is convex That means it bulges out. Convex, transparent structure, okay, that collects light from multiple angles. Okay, kind of like the security mirror in a convenience store, you know, the ones that are at the ceiling of the corner, right? It's a convex mirror. It allows you to see from a wide angle. Okay, that's essentially what the cornea does, collects light from a wide angle. Okay, and that is the most anterior portion of your sclera. Okay, so 3 layers, right? Sclera is the outermost layer. Underneath the sclera is going to be your choroid. Okay, it's the same word as choroid plexus that we learned earlier. Okay, choroid just means like vascularized structure that has a lot of blood vessels. Okay, so in this choroid layer, you will have blood vessels that help nourish the eyeball. Okay, eyeball is alive, so it definitely needs to have nutrients going to it, and most of it's done through the blood vessels within the choroid. And then the third layer is the retina. Okay, retina. The retina is where you will find the photoreceptors. Most of the photoreceptors are going to be at the back, right? No point putting photoreceptors here because they will never see light. Okay, the light comes in and then it's going to strike at the back, right? So that's the third layer. Sclera, choroid, and then retina. We'll write all these down later on. Okay, now located at the back of your eye, there is an area called the fovea. Okay, fovea. The fovea is where you would have the highest density of cone cells. Okay, cone cells are the photoreceptors that detects light, right? And it gives you high definition image. Okay, so whatever you're focusing on is going to be projected onto your fovea. So if you're looking at the computer screen or looking at your phone right now, that the image of the computer screen of your phone is going to be projected onto the fovea. Okay, now I usually do this demonstration in class, but I don't think it's gonna work here. But let me see if this is gonna work. Okay, like do you guys see the random math equation on the camera? Right, you might, right? Okay, so I am going to also put the anatomy stuff next to it. Okay, so if you focus on the math stuff on this page, okay, if you're looking at the camera, okay, you can look at the math on this booklet here, right? If you focus on that, the math is what's going to be on your fovea, okay? But as you're focusing on the math, you are aware that there are writing on the other booklet, on the anatomy booklet, but you cannot really make out what exactly is saying on it, right? You can only see what's on the anatomy book if you shift your focus to the anatomy book. So now if you look at the anatomy workbook, you can see the writing on the anatomy workbook, but now you won't be able to see the math, even though you're aware that the math writing is there, right? Okay, so in that illustration, I don't know if that actually works or not. Okay, it's the first time I do it on the camera. In that demonstration, right, it shows you that there's only one thing that can be on your fovea right now at a time. Everything else is going to be on your periphery. Okay, and when you're on the periphery, your peripheral vision, that's going to be picked up by the rod cells. Okay, so cone cells in the fovea, rod cells everywhere else. Okay, so that is that. Now there is the optic nerve that is going to take all these information from your eyeball to your brain. Anybody remember visual information? Which lobe does it go to? Which part of the brain does visual information go to in your brain? Rochelle said occipital lobe. That is correct. Okay, so it's going to go to the thalamus first. Thalamus will project that onto the occipital lobe. If it's information that is relevant. Okay, sometimes it's irrelevant information. It gets filtered out and then it won't get processed. Okay, in class, I usually at this point I would take a seat, and then the table at the classroom actually is very high. So if I sit down, you will only see, you know, like this above my chest. So I would sit down, and then I would ask the class. I would be like, you know, you have been staring at me for like an hour now, right? In lecture, I've been talking to you, teaching to you for an hour. Can anybody tell me what is the color of the belt that I'm wearing? Okay, and then people would have no idea. Okay, so that's an example of where you are seeing things, but you're not processing it, right? Okay, nobody cares what color the belt the teacher is wearing. Okay, your thalamus is going to filter that information now. It's irrelevant. Okay, we cannot process 100% of everything we see, right? We will get sensory overload, right? Some people can, right? You have heard of people who have like photographic memory, right? They see something and then they will remember it, right? They actually takes it all in, right? But for average person, the thalamus will filter out irrelevant information. Okay, now where the optic nerve comes out is going to be a blind spot. Okay, no photoreceptor on it, and you know, each eye is going to have its own blind spot. And you know, there are some blind spot tests that you can download online to see. Again, I cannot do this in a virtual environment with you, but it's good to find out your blind spot. Okay, a couple of other things. If you look at the front of the eye, you have the color part of the eye, right? That is the iris. Okay, your eye color is genetically determined, right? It usually matches with your hair color or skin color. So if you have lighter skin tone, then your iris is going to have a lighter color, maybe blue, maybe green. If you have darker skin tone, your eye color is going to also be darker, right? Maybe brown eyes. So it's because of a pigment called melanin, right? It's the same pigment that is found in our hair, in our skin, as they are in the iris. Okay. There is the lens here. Okay, the lens help focus the light onto the fovea. Okay, so the light would come in and the lens would bend it so that it would land on the fovea. Okay, if your lens deteriorates over time, right, then we can have a bunch of problems. Your eyeballs can also get too long or too short, and that causes people to have nearsightedness or farsightedness. Right? I already talked about this in the lecture, so you can review that yourself. Now, what's important to remember, to note here, is that the lens is actually held in place by these ligaments. Okay, you see all these things I just highlight. Let me see if I can rotate a little bit. There. Okay, so all these ligaments hold the lens in place. Okay, when the ligaments get tight, it will pull on the lens and make it thinner. Okay, when the ligaments are loose, then it will relax and the lens gets thicker. So let me repeat that one more time. Tight ligaments, thinner lens. Loose ligament, thicker lens. Okay. The ligaments themselves cannot change the tension. They cannot change the tightness. What changes their tightness are these ciliary muscle. Okay. So the muscle actually, you can see right here, it's connected to the ligaments. So when they contract, right, because it's a circular ring, when they contract, the muscle actually goes like this. It becomes smaller. Then the ligaments become loose, which would cause the lens to become thicker. So by changing the thickness of the lens, it helps you focus on things that are far away or nearby. If you're looking at something that's really far away, you want your lens to be thinner. Okay. If you are looking at something close by, like reading a book, right, you want your lens to be thicker. Okay. So if this is the classroom, what you'll be doing is like you'll be taking notes and then you'll be looking up, right, looking at the screen, and then you'll be looking down, copying stuff from the screen. So as you look up, your lens becomes thinner. And then when you look down and copy the notes, your lens becomes thicker. Okay. If a bus is really far away and you try to see what is the number on the bus, do we want the lens to be thick or thin? If you're thinking thin, that would be correct. Okay, so see, we don't have to consciously think about it, right? Your eyes, your nervous system automatically adjust the thickness and the thickness of the lens, right? As you shift back and forth, right? So that ability is called visual accommodation. Okay, as you get older, you will lose this ability because all these suspensory ligaments are going to lose their elasticity. And as a result, you won't be able to change the shape as quickly, right? So older people, when they try to read, they cannot really see it. So you see them like move their glasses and stuff like that, right? They actually need reading glasses to help fix that problem. Okay, so that condition is called presbyopia. Okay, so there is the overview of the eyeball, right? If you did not watch the recording, that is just like a 10-minute quick summary of the eye. So let's fill in the blanks. Identify the part of the eye in each of the following statements. Attaches the lens to the ciliary body. Okay, so the ciliary body is the muscle that controls the tightness of the suspensory ligament. Okay, fluid in the anterior segment that provides nutrients to the lens and the cornea. So as you can see, the lens and the cornea has to be transparent, and there is no way for the blood vessels to get to it. If the blood vessels were to go into the cornea and the lens, then we see the blood vessels in our field, right? And we obviously don't. So to get nutrients, there are actually fluids that fill up this gap right here. That fluid is called the aqueous humor. Aqueous humor. Okay. The funny water, some people say. The white of the eye. White of the eye is the sclera. Underneath the sclera is the vascular layer, which is the choroid. Vascular means it has blood vessels, right? Lots of blood vessels. Underneath the choroid is going to be the layer that has the rods and the cones, and we call that the retina. There's a gel-like substance that helps reinforce the eyeball. So here is that gel-like substance, right? So the eyeball doesn't collapse on its own. That is called the vitreous humor. Okay. So we have to drain the aqueous humor. If it builds up, then it increases the eye pressure. Same thing with the vitreous humor. If the pressure gets too high, it will compress the optic nerve. And it could lead to a condition called glaucoma, which ultimately causes you to become blind. The most anterior part of the sclera, cornea. Okay. Let's fill in this chart. Nearby object, like reading a book, okay? The lens, we want the lens to be thick. Okay? In order for it to be thick, the ligaments needs to be loose. And to make the ligament loose, the ciliary muscle has to contract. Okay? It's a little bit hard to, visualize this, but the ciliary muscle, as I mentioned, it's a ring. So when it contracts, it grabs onto each other like this and it gets smaller. Okay. So if you imagine, this is the ring. Okay. And then you have all the ligaments here, ligaments. And then you have the lens in the middle. Okay. So if the ring gets bigger, if the rings get bigger, then all these ligaments will get pulled on all sides, right? Okay, and then the lens will become thinner. Okay, So when the ring contracts, gets smaller, ligaments are loose, and the lens will be thick. Opposite, you want it to relax, so it gets bigger. And then the ligaments will get tight, which will then pull on the lens on all sides, making it thinner. Okay. So this is when you want to do nearby reading a book. This is far away. All right. Any questions? Let's see if we have any poll to do here. Let's see. Okay, let's do. Can we actually do this? Yeah, we probably can. Okay, let's do a poll for the first one. Go ahead. Oh, sorry. I always make this mistake. This is multiple answers. Okay, so select all correct answers. Go ahead. So the correct answer would be in the cochlea, which is the inner ear. That's for, you know, if you rotate your head, you feel that that's mechanoreceptor. And also underneath the skin, right, for feeling pressure. Okay, next one. Also, select all correct answers. Anybody else? There's only 5 responses. I'm gonna end it soon. Okay. So visceral nociceptors. Visceral are like deep inside, right? So angina, it's like heart attack, right? Or chest pain rather. Kidney stone, these originates from deep inside the body, from things that we don't have control over. So that's visceral. Cutting a finger, bruising from a fall, dislocating a shoulder rather, sorry. Those are somatic nociceptors. Okay, let's take a break. We will resume at 9:40, okay, in 5 minutes. Okay, see you soon. And we are back. Let's keep going. Next question. We are going to fill in this chart related to the structure. First one, retina. Okay. So the retina contains cone cells and rod cells. Okay. These are photoreceptors, right? Different types of photoreceptors. Cone cells are for creating sharp image and color vision. Okay. Three types. Red, green, blue. Okay. And if you mix different colors, you see different things, right? There's a color wheel in the lecture notes that you can see. It will show you when you mix different things, what color you will see. So for example, red and green together, if red light and green light enter your eyes at the same time, we will see yellow. Okay. If red and blue enter your eye at the same time, you will see magenta, okay, purplish pinkish color. Okay, rod cells. Rod cells are for peripheral vision. Okay, and for low light condition. Okay, that's the retina. Innermost layer, I should say, innermost layer of the eyeball. Any question? Okay.
What happened if the retina is damaged?
Yeah, so we could damage in various conditions, right? The most common one that you might have heard of is like retinal detachment, when the retina, like part of it tear off. Then you will lose, like there might be like a spot in your field of view that is not producing vision. Okay, so like you might see like a like if I'm looking at my workbook, right, there might be like a piece of it that I have no vision of. Okay, that's one possibility.
But would the cone cells be damaged?
The cone cells, well, for that part, right, that's detached, right, they wouldn't be functioning, right? So you will lose part of the vision, okay, for that location. Yeah. Fovea. Fovea is a special location on the retina that contains the highest density of cone cells. Okay, no rod cells here. No rod cells in the fovea. It produces the sharpest image. Okay, remember, whatever you are looking at, that moment, the thing that you're focusing on, that's the thing that's going to go on your fovea. Okay. I just realized another example I could use instead of the 2 pages of workbook I was trying to do. That was horrible. I'll never do that again. If you are on a laptop right now and you're looking at the screen, right? Okay. The screen is on your fovea. You are aware that there are letters on your keyboard. okay? But you cannot see what those keys are saying, okay? Because those, the keyboard letters, they are not on your fovea. They are on your peripheral vision. So those are picked up by the rod cells. Do you understand? Okay. Fovea is going to be processing the object of interest. Okay? We can only focus on one thing at a time. Ciliary muscle. Okay? So ciliary muscle is responsible for regulates the tightness of suspensory ligament, okay, which in turns adjust the thickness of the lens. Right? Ciliary muscle contracts, ligaments get loose, the lens gets thick. Ciliary muscle relax, the ligaments get tight, and the lens gets thin. Iris. Iris is the color part of the eye. Color part of the eye. Okay. And is not just to make our eyes look pretty. The iris actually changes the size of the pupil. Okay. If it's dark, you want the pupil to open up, right? So you can see, you can allow more light into the eye. Right? If it's bright, you want your pupil to be small, to constrict. Okay. So of course, the size of the pupil is controlled by your autonomic nervous system. If you remember from lecture 5, right? There is the sympathetic system, right? The flight or fight, and then there is the parasympathetic system. Okay, so the sympathetic system is going to cause your pupil to dilate. Okay, and then the parasympathetic system is going to cause your pupil to constrict. But the most direct way to change the size of pupil is with light. Okay, you probably seen this on TV shows or movie, right? Some person had a terrible accident. They're lying on the bed, and then you see this doctor come and take out like a little flashlight from their pocket, and then they lift the person's eye and then shine into it, right? And then you can see the pupil constrict, right? Okay. If the pupil does not respond to the light, then there might be some neurological damage. Okay. Also, fun fact: the size of the pupil on one eye is not necessarily connected to the other. Okay, so if one eye is stimulated by light, right, and this one constrict, the other one is not going to constrict necessarily. Okay, so if you wear an eye patch, okay, and then you go out and it's really bright, pupil on here is going to constrict because of the bright light, but this one is kept in the dark, the pupil will be dilated. Okay, so they're not necessarily like, you know, in sync with each other. Okay, aqueous humor. What is aqueous humor? Aqueous humor is the liquid that is at the front of the eye, right? Okay, so this one is going to nourish the cornea and lens. Okay, it contains nutrients. It also helps reinforce the shape of the eyeball. Okay, the front part anyways. And we want to drain the fluid So drains through the sclerovenous sinus. Okay. Venous sinus is a place to drain stuff. So we have one at the sclera. That's why it's called sclerovenous sinus. We have one in the brain, remember? The durovenous sinus, because that's formed by the dura mater. Virtus humor takes up the entire space, the posterior part of the eyeball. It's a gel-like substance. Gel-like substance that fills up the posterior cavity of the eye. Also reinforce the shape of the eyeball. Okay. Any other question about the eye in general before we move on? All right. I'm going to fast forward to this diagram. I know I did it in class already, but it's such like a confusing thing that it's probably worth talking about it one more time. Okay. So this is a transverse view. Okay, if you cut the plane like this, and then you're looking at it from the top. Okay, so you see the eyeballs. Okay, this is the right eyeball, the right eye, left eye. Okay, so we can think of the eyeball as divided into two halves, the right halves and the left half. Okay, so each eyeball has a right half and a left half, right? Even the left eye has a right half. Okay. Now we have something called the field of view, FOV, right? So FOV, field of view. Right? So let's say there is here one person, and I'll show in red. This would be the right field of view. When the light bounces off the person on the right field of view, it will enter your eyeball and strike the left side of your eyeball. Okay. So first thing to know, left half of both eyeballs will be receiving information about your right field of view. Okay. Left half of both eyeballs receives light from right FOV. Is that okay? All right, we're gonna have a blue person here. This is left field of view. And then you can kind of write the sentence yourself. The left field of view will go to the right half of each eyeball. Okay, and obviously you're gonna have some overlapping right in the middle. Things that are in the middle will be processed by, both half. Right, but let's keep it simple, left and right here. So where do they go? Well, this is the optic nerve. Optic nerve. Okay, the left one, of course. There is another optic nerve. And what's going to happen is the right field of view information, let's focus on the left eye here. That information, it's going to travel along the optic nerve. It's going to go towards the midline of the body. Okay. And then for the left eye, it is going to continue on the same side of the body. Okay. On the same side of the body. So this is outside of the brain, which is why it's called the optic nerve. And then past this point, it's going to be inside of the brain now, which we will now call the optic tract. Remember, tract and nerve are a bundle of axons. The difference is whether it's found in the PNS, outside of the brain and spinal cord, or inside the brain. Right? So optic nerve, optic tract. And then it will go to the thalamus. Okay. All these are on the left side. Similarly, on the right eyeball, right? The right field of view information on the left half of the right eyeball will travel along the optic nerve on the right side. But instead of staying on its own side, what's going to happen is it will cross the middle and it will join the optic tract right here, and it will continue to the thalamus. Okay, so for the left eye, the information on the right field of view, represented by the red, will stay on the same side. Whereas on the right eyeball, information from the right field of view will cross the midline. Okay, so together, the right field information will go to the thalamus, and then it will go to your right occipital lobe. Left occipital lobe, rather. Left occipital lobe. Okay, so what does that say? Well, the left side of the brain actually receives information from both eyeballs. Okay, sometimes we get, too ingrained with this notion that the right half of the body, it controls the left half. The right half of the brain controls the left half of the body, and then vice versa, right? But there are exceptions. So in this case, both eyeballs will send information to the left brain, and they will send information to the right brain as well. What's different is that the left lobe, the left occipital lobe, will process your right field. Okay, so it's the field that goes to the opposite side of the brain. Okay, but remember, both eyeballs are going to be processing information related to the right field. Okay, so this location in the middle, where the optic nerve crosses each other, this is called the optic chiasma. Okay, optic chiasma. Okay, and it's located just in front of the hypothalamus. Okay, let's finish with the blue stuff. Very, very similar situation here. If I can just find the blue color, I think it's this one. Yeah. So the left field of view entered the right half of the eyeball. For the right eyeball, it will stay on the same side. For the left eyeball, it will cross at the optic chiasma and join with the other fiber, and then it will go to the occipital lobe. Any question about this concept? You can see how it is quite confusing. No question? All right, let's go through these questions then. First one. Right optic nerve carries information from which field of view? Okay, let's take a look. The right optic nerve. Which one is the right optic nerve? Right optic nerve is right here. Okay. What kind of information does it carry? Well, you can see both color here, right? So it's both left and right. Left optic nerve carries information from which FOV? Same thing, both. The right optic nerve, the right optic tract carries information from which FOV? Right optic tract is right here. You can see it exclusively is blue, which represent our left FOV. The left optic tract would be the right FOV, or red. Okay, next one's a little bit more involved. The right optic tract, which is this one, carries information from which part of the eye? If you trace the blue all the way back to the eyeball, it is the right half of the eye, of both eyes. Whereas, keep on spelling right wrong. Whereas the left optic tract is going to be left half of both eyes. Okay. You can see how the question on the test could like mess with you a little bit, right? Because there's so much going on. My suggestion is you draw this diagram a couple of times, see if you understand it, you know, have a nice diagram next to you on the test. Then you can trace the color easily. Okay. See if any polls on that. Right here. Okay, let's see. Number 7. Please try this. Try to do it without looking at the diagram, would be nice. All right. Almost all of you got it right. That is correct. Okay, D is not correct. Visual information from the right field of view, that is carried by the left optic tract. Again, you can just trace the color, and you should get the answer. Okay, that is that. Let's do this one as well. Okay, myopia, which is nearsightedness, and then hyperopia. So this is basically the like most common eye condition, right? So what happens in each of these? Okay, so for myopia, nearsightedness, right, you would have difficulty seeing far. Okay, the name tells you what you can see. Okay, so nearsight means you can see near, but not far. Hyperopia, farsightedness, okay, you can see far, but you would have difficulty seeing nearby objects. Okay, close by, close by. Okay, far away objects. For nearsightedness, the eyeball is too long. For hyperopia, eyeball is too short. Exaggeration, of course, like no one's eyeball is going to look like that. Yeah, but it helps you remember. So this is the pupil at the front. Okay. Actually, you know what? Let's just draw the lens. Okay. So too long, too short. Converging points of light rays, because the eyeball is too long. So what's going to happen is the light comes in, and then now they are going to converge. They're going to come together too soon because the eyeball is too long, right? It's like the projector, right? Okay. In class, we would have the screen here, and then we would have the projector, right? And then, you know, you will see the PowerPoint slide on the screen. If I move the screen further back, then the image will be blurry, right? Because the light rays are focusing in front of the screen. So in this analogy, the screen is like the retina, okay? Because you're moving the retina further away since your eyeball gets longer. The focal point is in front of it. So in front of retina. Okay. So how do we fix it? Well, to fix this problem, we are going to wear a diverging lens. A diverging lens is going to spread the light rays out. Diverging lens, which will spread the rays out a little bit before it enters our eyes. So we'll just spread it by the right amount such that it will converge on the retina again. Okay. On the other hand, for hyperopia, the light rays come in and then they would be converging behind the retina. Okay. And you would need to wear a diverging lens to spread, converging lens to make the light rays come together sooner, okay? So that it will be right on the retina again, okay? Another example of a converging lens is a magnifying glass. So people who have hyperopia, sometimes you can see their eyes are bigger, right? If they wear the converging lens, because it's just like a magnifying glass. Okay. Which I remember, does anybody know like the TV show Survivor? Survivor, anybody watches that like before? It's like a bunch of people who go on this remote place and then they have to play games to eliminate each other. And then I remember one year I watched an episode where each of these contestants can bring like one object with them, right? And then this person, you know, the team, nobody bought anything to start a fire. And then he's like, oh, don't worry, I have my glasses. And then I could use my glasses to start the fire, right? And then what happens is he tried, but then it didn't work, right? And then I remember I was laughing because he actually have a nearsightedness, okay? So his lens is a diverging lens. Diverging lens, you cannot start fire with them because diverging lens spread the rays out, right? Rather than focusing it like a converging lens, okay? So only magnifying glass can focus light rays and potentially start a fire, okay? So if you're stranded on an island and you want to start a fire, you have to have farsightedness and wear the converging lens to do that, not the other way around. So that is that for the eyeballs. Okay, no more eyeballs. Any last minute question for eyeballs before we move on to the ears? Okay, quick overview for the ears before we fill it in. So we have the external ear, which contains like the visible part of the ear. We call that the pinna, P-I-N-N-A, pinna. This is the ear canal, right? Where the sound is going to go in. You know, when you use your Q-tip to clean your ear, you're basically cleaning the auditory canal. Okay, that's also part of the external ear. And then at the end of the auditory canal is going to be your eardrum, okay, which is this part right here. Okay, fancy word for eardrum is the tympanic membrane. So the tympanic membrane will move as a result of sound waves, and it basically converts sound energy into movement. Okay, it's the opposite of what a speaker does, right? Okay, speaker creates movement, right? And then that movement translates into sound. So nowadays, right, a lot of people check temperature, right, using those ear things. If you have kid, you might have one of those ear thermometer. Basically, it checks the temperature of the tympanic membrane, checks the temperature of your eardrum. So on the medial side of your eardrum, it is connected to these bony stuff. Those are called your ossicles, okay, tiny bones. There are 3 tiny bones in your inner ear, okay, in your middle ear rather. Okay, they are called the malleus or the hammer because it looks like a hammer. The incus or the anvil. And then there should be a stapes here, which I'm not sure why it is not shown. Okay, there is going to be a third one here that looks like you know, the thing that you put your feet on when you ride a horse. I don't. Most people haven't ridden a horse before. I haven't ride a horse, but then from TV you see people put the foot in one of those things, right? It looks exactly like that. Okay, but for some reason it's not showing here. That's called the stapes or the stirrup. So these bones that will help amplify the sound. Also in the middle ear is this tube called the Eustachian tube, also known as the auditory tube. Typically, this tube is closed. It will open when you swallow. Right? It connects to your nasopharynx, and anytime it opens, it releases pressure that builds up in your head. It helps you equalize the pressure inside the head with the external environment. So the best example of this is if you are in a plane, the plane is going up, you feel your ears getting plugged. If you're in an elevator that goes up really, really high, sometimes you feel your ears getting plugged as well. So as we go up in elevation, the pressure outside drops, and then the pressure in your head builds up. And when you swallow, it opens up the Eustachian tube. That extra pressure comes out, and then it equilibrates with the outside. There is going to be the inner ear here, which contains this snail shell looking thing. That is the cochlea. Okay, inside the cochlea is where you will have mechanoreceptors that helps you pick up sound. Okay, and then over here there are these semicircle structures. I wonder if I can delete some. Oh yeah, there we go. Okay, if I delete a bunch of these things, okay, you will see that there are 3 semicircular tube here. They're called the semicircular canal. So one of them, like this one, that's called the anterior semicircular canal. That one is parallel. So like if you think about where you are, right? If you're in a room, right, you would have a wall behind you. You would have a wall to your side, okay, and then you have the floor and the ceiling, right? Okay, so the anterior semicircular canal, the one that I'm pointing at right now, that's going to be parallel to this plane, okay, parallel to the wall that is on your side, okay? So any motion that's parallel to that plane will be detected by the anterior circular canal. So if you nod your head, if you say yes, nod your head, then the semicircular canal is going to be detecting that, okay? There is going to be the posterior semicircular canal. That one is parallel to the wall behind me or to this piece of paper, okay? So that will be activated when you tilt your head to the side like this, okay? And then the one down here, right? That one is parallel to the floor, right? Or to this plane, right? So that's going to be activated when you shake your head like this, okay? When you say no. Okay? So that detects head movement. Connecting the cochlea and the semicircular canal is going to be this structure here called the vestibule. So in the vestibule, it is responsible for detecting acceleration. So if you're falling or if you are speeding up or slowing down in a car, that feeling of speeding up, slowing down, or falling is detected by the vestibule. All this information is going to travel through the vestibulocochlear nerve to your brain. Okay, so that's pretty much it. There's also a round window and the oval window here, but I'll explain in a bit. Okay, let's go back. Ossicles. Okay, so you have 3 ossicles. Okay, from lateral to medial, we start with a malleus, incus, and then stapes. Their common names are hammer, anvil, and stirrup. Okay, resembling like real life objects. The function of the ossicle is to amplify sound. Okay, they transfer the movements from the eardrum or the tympanic membrane to the oval window. Okay, so the amplification is about 20 times. Okay, let me briefly explain the oval window and the round window. Okay, so this is the cochlea. Okay, and then you have your vestibule and then your semicircular canal. Okay, but let's focus on the cochlea. The cochlea is a snail shell looking thing. It's actually a bony structure and it's filled with fluid on the inside. Okay, so there is one membrane here and then another membrane down here. Okay, let's do them with different color. One membrane on top and then one membrane down here. Okay, you can see them more clearly if I go down to this ear diagram here. Okay, so oval window, round window. Okay, I know I drew it on here. It's just easier to draw rather than the whole thing. Okay, so 2 membrane over here. And let me go back to this diagram. Okay, so I'm gonna roll this out for you. I'm gonna unroll it into a long tube like this. So if you just take the snail shell and then unroll it, that's pretty much what it's gonna look like. So on the inside, there are actually 3 separate channels. Okay, so there's a top channel like this. It's top channel, bottom channel, and I'm just going to draw the middle one here to show you. Okay, know that there is also top chamber and the bottom chamber, and it's filled with fluids. Okay, so you saw the malleus, which looks like a hammer, and then the anvil, and then the stapes, which was missing, right? So the stapes looks like this. It looks like, again, the thing that you put your feet on if you're riding a horse. So the stapes is going to be connected to the oval window. Okay, so when the eardrum moves back and forth like this, the ossicles will move, and then the stapes would press into the oval window. Okay, and that pushing on the oval window will create pressure that would now start to move through these fluid. Okay, so the purpose of the oval window, is to transfer the movements from the stapes into pressure waves that will be transmitted in the fluid inside the cochlea. Okay. Now, depending on how much energy the sound has, okay, this wave is going to turn around. It's going to turn around. Okay. If it's a lower pitch sound, then it's going to go further down before it turns around. Ultimately, it's going to turn around. Okay. And when it does turn around, that excess pressure, it's going to cause the round window to bulge out. So basically, this is a confined space that is filled with liquid. So when you press in on the oval window, when the stapes press in on the oval window, the round window will bulge out to accommodate for that extra pressure. When the stapes goes back out, then the round window will go back in. Okay, so basically, these two windows move in opposite direction to ensure there's no excess pressure building up on the inside. Okay, so the round window, what does the round window do? As the oval window gets pushed in by the stapes, the round window will move outward to relieve the excess pressure. Okay. Now, in the middle where it turns around, there is a structure called the organ of Corti. Organ of Corti. Now, without going into too much detail, because you don't have to know it, the organ of Corti is going to have hair cells, which is the mechanoreceptors that we found within the ear. Okay, so a hair cell looks something like this, okay, with hair in it. So there is going to be hair cell here, all over the middle. Okay, so you can think of these hair cells as like the keys on a piano. Okay, depending which one gets pressed, then you're gonna get a different sound, right? Okay, so depending on where the wave turns around, those hair cells will be stimulated. Okay, so typically speaking, the lower pitch is gonna go further in and then stimulate these hair cells. And then the higher pitch will stimulate the ones that are closer to the ear. Okay, that's why as we age, especially if you listen to your headphones a lot, you will damage the hair cells that are closer to the outside. And then when you get old, like when you're in your 40s or 50s, you start to lose the ability to hear high-pitched sound, okay? Because these hair cells are all damaged, okay? So that's basically what's going on, all right? So I'm just going to skip to the last one here because I just talked about the organ of Corti. So it contains hair cells, which is a type of mechanoreceptor. Okay. And their function is, I should say they are located in the cochlea. Okay, and their job is to transfer sound, auditory information, to the cochlear nerve. Okay. All right, let's fill in a couple more. Semicircular canal. What is the semicircular canal and what does it do? We talked about it already. Okay, semicircular canal detects. They detect head movement. Okay, there are three types. There is the anterior one, anterior semicircular canal. That's the one that is parallel to this plane. Okay, parallel to this plane. So when you nod your head, nod your head. Okay. There is another one, the lateral. Lateral is the one that's parallel to this plane. So when you shake your head, okay, when you shake your head side to side, shake your head side to side. And then there's the posterior one, the one that's parallel to this plane. Okay, so when you tilt your head to the side, okay, tilt head to the side. Okay, so it detects head movement. Okay, or it doesn't have to be your head. Like if your whole body rotates, right? Like if I rotate on my swinging chair right now, okay, like this. Okay, rotation. So tell me, if I rotate like this, right, which semicircular canal is going to get stimulated? Is it the anterior, the lateral, or the posterior? Anybody want to answer that? Rochelle said lateral, and that would be correct. Okay, lateral. Very good. Eustachian too. It equalize pressure inside the head with the environment. Okay, normally closed. But will open when we swallow, during swallowing. Okay. Tympanic membrane is just eardrum, fancy word for eardrum, and it converts sound energy into movement. Okay. Connected to the malleus on the medial side. I'm going to zoom out a little bit for anyone who needs to do a screen grab. Any questions? Okay. Any more polls? Let's see. Okay, this is just another one. Let's see if you guys can do that. This is a pirouette. Okay, I only have 3 people answering. The rest of you unsure? Okay, so basically there are 3 axes, right? The X and the Y and Z. I think of it as like the corner. This would be like the corner of the room, if you would. And then, this would be the wall behind me, the wall on the side, and then the floor. Okay, so basically, if you think about a person like this, there's the person standing here. Okay, a pirouette is basically a rotation on the z-axis, on the vertical axis. So going like this. Okay, so that's going to trigger the lateral semicircular canal. Okay, what kind of movement would trigger the posterior semicircular canal? Anybody? Okay, so rotation on the z-axis is going to give me lateral stimulation. What about the posterior? What kind of motion? Which axis should we rotate in? Anybody? Yeah, Rochelle said cartwheel, and cartwheel would be rotation in the x-axis. Okay, so for those of you who don't know what a cartwheel is, cartwheel goes like this. Okay, and then you rotate you rotate like this. Okay, like cartwheel. Okay, so that's rotation on the x-axis. That would stimulate the posterior one. Okay, and then for the anterior, it is going to be a somersault. Okay, so you rotate on the y-axis. So like if you go like this and rotate, I don't know if that makes any sense. But on the test, if I write a particular word, I will show you a picture just so you would know which one I'm talking about. All right, good. Okay, this is a very important diagram. I hope you guys have a lot of colors, okay? Because this diagram will not make sense if you don't have colors. Okay, so please get some colors. At least 5 or 6 different colors if you could, and then we'll do this together. Okay, so just to reorient yourself, just to help you remember what's going on. This here, right? What lobe is this? That is the frontal lobe, okay? Frontal lobe. Here is the parietal lobe. What page is this on? This might not be in the workbook. If you got the workbook from the bookstore, I do apologize about that. But if it's in the study guide that I posted, okay. If you don't have this, you can just quickly draw a brain. Okay, so something like this would work. Okay. Basically, you need to have the frontal, parietal, occipital, and temporal. Okay. Frontal, parietal, occipital, temporal. Okay, so let's go back to this. This is occipital lobe. And then this would be temporal lobe. All right. Cerebellum here at the back. And you can't really see it here, but it's inside and it's important. So I'm gonna do like a halo outline for it. That is going to be our thalamus. Okay, so those are all the parts that we need. Okay, so let's start with eyes. We're going to write down the senses and we're going to write down the type of detector. So for eyes, it is, of course, for visual, right? Visual information. The type of receptors. What kind of receptors do we find in the eye? Anybody? We talked about 5 types of receptors. What kind of receptors do we find in the eye?
Photo.
Yep, thank you. Photoreceptors. Okay, the rods and the cones. Okay, so here we go. Watch. The eye, right? This visual information will go through the optic nerve past the optic chiasma and then the optic tract, which will then go to the thalamus. Okay. So from the thalamus is going to process the information and see if it's worth passing it on. And if it is, then from the thalamus, it is going to go to the occipital lobe. Okay, remember along the path, there are many things that it will hit, right? I just mentioned a few, the optic tract, optic chiasma, optic nerve. But that would be the sensory pathway for visual information. Okay, the nose is for smell, fancy word for smell. It's like sight, we call it visual, right? So for smell, we call it olfactory. Olfactory. Okay, I did not really talk about the olfactory in the tutorial today, but it was in the recording. Anybody know the type of receptors for olfactory, for smell? No? Okay. Chemoreceptors. Okay, so your receptors that pick up different scent molecules, right? Okay. So for smell, what lobe does it go to? Anybody? We learned that last week. Okay. It's going to go to the temporal lobe. Okay, temporal lobe right here. It's going to bypass the thalamus. Is the only sense that doesn't get filtered by the thalamus. Okay, it goes directly to the temporal lobe. Temporal lobe. Okay, this is a taste. Fancy word for taste, gustatory. Gustatory. What kind of receptors do we have for taste? Also, chemo. Yeah. So we would have receptors for tasting sweetness, right? Those are glucose receptors. Salty, those are sodium receptors. You might have receptors for like bitterness, right? For acidity, different chemoreceptors for that. Now, also in the tongue, we also would have thermal receptors as well. Okay, not for the taste, but for like temperature, right? You know, you can taste, you can feel temperature with your tongue, right? When the food is too hot, you will feel that. You're eating ice cream, it feels cold, right? So those are thermal receptors, as well as mechanoreceptors. For like feeling textures, okay? So where is it going to go? Well, it is going to go up through part of the brainstem, and it will go to the thalamus first. Now from here, it goes to 2 places, okay? Related to the taste, like the sweet, sour, bitter stuff, that's gonna go to the insula. Okay, the insula is behind the frontal lobe and the temporal lobe. If I take two forceps and I pry it open, like as if like opening a curtain or something, then I would see the insula inside. So that will go there. All the other information related to temperature, pressure, texture, all that stuff, those will actually go to the primary somatosensory cortex. Okay, so I don't know if you remember from last class. This is going to get really busy, this diagram, but let me try to shade it out for you. This part of the brain, okay, that is the primary somatosensory cortex. And over here, that's the primary motor cortex, right? That's for all voluntary control. Has nothing to do with what we're talking about, but I just put it in for your reference. This is primary somatosensory cortex. It's for all the somatosenses, right? Okay, bodily senses. So anything you feel with your skin, with your muscle, right? All that stuff goes there. Okay, so last class I told you it's separated into different zones: the body, the hand, right? The eyes, the head, right? And down here, that's where the tongue is. Does everybody understand why this path splits into two separate pathways? Okay, the hand, not necessarily just the hand, but like the entire body, right? Covered by anywhere covered by skin. This is somatosenses. Okay. So the kind of receptors we have would be mechanoreceptors for pressure, thermal receptors, right? Nociceptors. And then like deeper into the skin, we also would have like in the muscle and stuff, we would have something called proprioceptor. Okay, we didn't really talk about proprioceptors, but I'll tell you now. It basically detects like stretching of the muscle, stretching of the tendons. How much they stretch gives information about how your muscles are moving, right? So that's vital information when it comes to things like balance, maintaining balance and equilibrium and all that stuff. Okay, so for this one, let me see the pink one right here. Okay, it's gonna go up through the ascending tract, okay, through the brainstem. Okay. Part of it might go to the cerebellum. The rest is gonna go up to your thalamus, and then from the thalamus is going to go to your primary somatosensory cortex. Okay. It's a very busy diagram. I'll let you take a screenshot after. This is the cochlea. And cochlea, vestibule, and semicircular canal. Okay, all these things. So together, what do they do? Equilibrium, right? Balance. And to do that, you would need to have mechanoreceptors. Okay, the hair cells that we talk about, right? Running out of color here. So where do they go? Again, up the brainstem. And it's going to go to the cerebellum. Okay, and some of these information will also go to your thalamus, and then to your primary somatosensory cortex. Okay, almost there. For hearing or auditory, we would need mechanoreceptors. Oh, sorry, cochlea should not be here. My bad. The cochlea should be right here. Right, the cochlea. For balance and equilibrium, it's just the vestibule, which is this part for acceleration, right? And the semicircular canal. Sorry about that. And then the hair cells, right? The mechanoreceptors in the cochlea. Those are gonna go up. Thalamus again. And then it's just gonna go to your temporal lobe. Remember, in the temporal lobe, there is the Wernicke's area. That's for interpretive area. It's your interpretive area, right? And helps you understand what people are saying and helps you understand what you're reading. Okay, so that is that. I know it's kind of small, but if you want to take a screenshot picture, you could do that. Are we okay? All right. Application question. You stare at a bright red screen for 30 seconds. Bright red screen for 30 seconds. What color will you see? What color will you see if the screen is switched to each of the following color? Okay. If it's switched to white, what color will you see? Okay. So let me just help you refresh your memory. Okay, so like you have three kinds of photoreceptors, right? You have the red, you have the actually this is not going to work because this is like coloring color. What we need is like light color wheel. Does that work? Oh, there we go. Can I copy this somehow? Right here. Okay. So I'll put this here and then I will let you answer the question. Okay. You stare at a bright red screen for 30 seconds. What color will you see when you switch to a white screen? Okay, please type your answer in the chat box. What will you see? Anybody? Should we try this? Let's try this. Let's try this. Assuming you guys are looking at the screen rather than just, listening. I know some of you said you're working and you're just listening to me. But if you are not, let's try this. And then maybe you will see what I mean. Okay, I'm gonna give you the bright red screen. Okay, please stare at it. Okay. As you're staring at this very red screen, okay, the red cone cells are working really hard. Okay, you have three kinds of cone cells. You have the red, the green, and the blue. and the red ones are working hard to register this bright red color. Okay, the blue are not doing anything at this moment. The greens are not doing anything because they're not needed. To see the color red, you only need to have the red cone cells. Okay, so I hope you have been staring at this for a long time. I'm going to switch to a white screen in 5, 4, 3, 2, 1. And I want you to blink your eyes a couple of times. And then I want you to type in the chat box and tell me what kind of color you see. Okay. Anyone else? Really? You guys see blue? That is.
Yep.
Oh, like, okay, like I see what you mean. Okay. Yeah, okay. So like a lot of us are not artists, but do you mean like this blue, or like do you mean like this blue, or do you mean like this blue?
The
Cyan,
blue.
right? Yeah, okay. that makes more sense. Okay. So what's going on, right? What's going on? Well, on a white screen, how do we see white? Okay. We see white when all 3 of the photoreceptors are activated. Okay, so in order to perceive the color white, we need the red and the blue and the green to be firing. When I say firing, I mean transmitting action potential at the same intensity. Okay, so what happens is when we were staring at the red screen, the red cone cells I said was working really hard, right? So when we switch to the white screen, the red ones are like tired out. Okay, they don't want to do it anymore. They have been transmitting so long. Now they need a break. Okay, to put it in scientific terms, some of them are like in their refractory state. They're not ready to transmit signal again, right? So on the white surface, the blue and the green are firing stronger than the red. Okay, and as a result, when blue and green are fired at the same time, you see the color cyan. Okay, so that's why that's the answer. Does that make any sense? Hopefully it does. Okay, what if we switch to a green screen instead of a white screen? Okay, what color will you see? Well, to see green, you only need the green receptors, right? Okay, we don't need any of the red receptors, right? So on the green screen, you will have no problem seeing the green. Okay. What about the yellow screen? Now, this one is the one that we'll see if you understand all along what I'm saying. Okay, if you stare at a red screen for a long time and then you switch to a yellow screen, what color will you see on the yellow screen? Green is correct. Green, right? Green. Because to see yellow, you need the red and the green, but the red is tired, right? Tired out from the previous screen. So only the green will be transmitting the signal on the yellow screen. All right, I hope that makes sense. If you see something like that on the test, I hope you'll get it.
So how come when you look at the green screen after the red, it's just the green?
Because to see green, we don't need the red receptors, right? It doesn't matter that the red was tired because I don't even need them, right? So the green screen will still appear to be green.
Okay.
Yeah. If we look at a blue screen, right, it will still be blue. Okay. If we look at a magenta screen, that would also be blue. Yeah, because anything that involves the red, they're going to be tired out. Okay. All right. One last question, and then we're done. David went for his yearly eye examination and was informed that his intraocular pressure, just means pressure inside the eye, was slightly elevated, okay, a little bit higher. The physician expressed concern over this condition and noted that if the condition got worse, eye drops would be needed. What is wrong with David's eye and what are the possible consequences of this condition? Speculate the function of the eye drop. Okay, so I mentioned earlier that if the eye pressure keeps on going up, it might lead to a condition called glaucoma. Okay, usually it happens in like older people, like you might even know someone who's has glaucoma. Okay, and the glaucoma is as a result of a damaged optic nerve. The high pressure is going to be pressing on that optic nerve, eventually damaging it, causing it to no longer be able to transmit information to the brain. Okay, so once that transmission is no longer there, you won't be able to see. So you will start losing your peripheral vision, and then eventually you will go blind. Okay, so the function of the eye drops is to lower the eye pressure. Okay, you can lower the eye pressure in a couple of ways. There are these blockers in these special eye drops that will slow the production of the aqueous humor. There are these drops that would encourage drainage. Okay, so whatever the mechanism is, the key idea here is to lower the eye pressure, right, and ease the tension on that optic nerve. Okay, that's it. All right. Now, next class will be quite packed because we have to do the tutorial for the digestive system. We have to take up the lab, and we have to do a little bit of a review. I'll post a review sheet probably like later this week, maybe Friday. And then you can work on it over the weekend. Now, I receive an email saying that my Centennial will be down on the weekend. It should not affect eCentennial, but just in case it does, download everything you need over like before the weekend and just have it on your computer. Okay, that way you will still have access to it. If you for some reason cannot find stuff, email me. I'll personally send you the files. All right, that's it. Thanks so much. Have a good week.
Professor.
Yeah.
With this glaucoma, the eye condition, it runs in my husband's family. So, but one of the stuff that they did was the laser to put the holes in the eyes.
Yes, which improves the drainage, right? I believe that's what the purpose is. Yeah. Okay.
Okay.
All right.
Thank you.
Thank you.